Thank you, everyone, for joining us for this wonderful conference. This is the first conference for the year for VJON. We've done multiple conferences over the years, but what we've got at the moment is a very serious situation from a scientific point of view, where the virus is still circulating, it is still doing damage, and there are not enough people trying to find answers. What we've got here is a group of global experts sharing ideas, sharing their research, and critically, towards the end, after each speaker has their presentation, they will be discussing it with the panelists. After that, each speaker in turn, and then at the end, we'll have a roundtable discussion, which can sometimes be the most valuable thing. And at that point, we can also answer some of the questions that you have. So I appreciate all the people who are already here with us, and we look forward to sharing with you wonderful ideas about what is going on and what can be done. So without further ado, I'll get all the speakers to just say a quick introduction for themselves so that we get to know who they are. And so we'll probably start with Bridget, since she's going first, and then Rezia, then Sabine, and then we'll get our two panelists, Shankara and Marcus. So Bridget, just go ahead and say hello to our audience.
Yeah, hello all together. I'm happy to be here. My name is Brigitte König, and I studied biology, chemistry, and medicine, and I'm a professor of medical microbiology and infectious diseases. And in addition, I have a small company which does specific analysis in direction of mitochondria and infectious diseases.
Wonderful, thank you. And Rezia?
Hello everyone, I'm Risha Pretorius. I'm from Stellenbosch University in South Africa. I'm also affiliated to the University of Liverpool, and I am a physiologist and work in the field of coagulation and microclotting. And of late, long COVID is one of my main focus areas.
Wonderful. And Sabine?
I'm Sabine Hazen. I'm a gastroenterologist by trade. I started my practice as a GI doctor and kind of went into the clinical trial route of doing clinical trials for pharmaceutical companies. My forte in GI and my expertise was in fecal transplant, intestinal microbiota transplant. When that showed a lot more than improving C. diff, I got curious and opened up Progenobiome to understand the microbiome and disease. And I'm spearheading 61 clinical trials on the microbiome and disease.
Excellent. Thank you very much. And Shankara?
Hi, everyone. I'm Dr. Shankara Chesi. I'm a general practitioner from Port Edward, South Africa. I'm a natural life science biologist and have been a frontline COVID doctor. My, I think, sentinel finding was that we're having an immune response with COVID rather than a virus causing all the pathology we're seeing. So, yeah, very interested in what's going to be discussed today. The sentinel things centering around the pathophysiology of what's causing all the long COVID and long vax injuries.
Excellent. And Marcus?
Hello, my name is Marcus Glotz. I am the founder and director of the world's first purpose-built long COVID, post-vac, and chronic illness clinic. And here in Cyprus, in Larnaca, we're focusing on offering the largest possible German high-tech portfolio of treatments at accessible prices. And what we do is we're working a lot with scientists around the world to make the latest treatments early accessible.
Excellent. Thank you very much, everybody. And as you can see, we've got quite an audience all over the world who are already with us. And I'm sure as we go along, the numbers will rise. So thank you, all of you who are here. I'm seeing all the comments, and as best as possible, I will share them in the discussion if we have time, but mainly probably towards the end.
Many functional doctors have protocols for improving mitochondrial function and switching the energy production from anaerobic glycolysis back to aerobic glycolysis with high-dose thiamine using fat-soluble variants, TTFD, benfotiamine, activated B complex, magnesium and potassium, selenium and molybdenum. Given the extent of the damage to the mitochondria, is this a viable solution, or do we need to remove the spike protein from these cells first?
Yeah, like your first question, I think it's both which we should do. Sure, we can start with destroying the spike or removing the spike protein, but if the tissue is need also all minerals to recover. Yeah, this must go a little bit in parallel. Yeah, sure, you can start first with the spikes, but then you have to look at the recovery of the cells. But I think a lot of people say Coenzym Q10, NHD, Nicotinamid dihydrogen, whatever. But we have to do a lot, not only one substance. Yeah, because if you really look at the biochemistry, they all interact each other. Yeah, and if you only look at one, you have problems at another position. Yeah, and then also you must look whether you have perhaps to remove the damaged mitochondria and to go into the mitogenesis, also biogenesis, to amplify or to regenerate the good mitochondria. Also it's not just one substance which you should give. Yeah, it's and then it's also one important is to look which biomarker or which biomarkers show me I'm on the right way. Yeah. Sure, one simple is to look that the spike protein is disappearing or destroying. Sure, that must be, yeah.
Yeah, that's why we keep DHL so busy with sending samples to you every day.
Yeah.
Wonderful. Thank you very, very much. Appreciate that, Rezia. And before I go any further and bring in the our panelists, I just wanted to take a moment to remember our guests who are with us, who are busy commenting. And I'll be trying to share some of the questions after our panelists have been able to share some their own questions, but I'll be looking out for questions from the audience as well. So let's get straight into our panelists then and see what questions they have to ask.
Perfect. Rija, thank you very much for this. Let me also congratulate you, of course, Dr. Koenig as well. Phenomenal work. Just your last paper, absolutely love it. And thanks for bringing the biocode tests to life with Yannick. Our patients and our clinicians here, we love it. Just the detail that we get now is amazing. Now, the biocode test, the flow cytometer, and the PET scans, is that the future of microclot testing in your eyes?
Yes, definitely. Right in the beginning, we looked at the microscopy. So that is our team's main research focus. We soon realized that microscopy is not useful for large-scale clinical utility. It's easy in a research setting. It is very difficult to do it on a large scale. So we crowdfunded via kernels, and many clinicians and patients contributed. We got our own imaging flow cytometer. We patented the method, and Biocode is distributing it to all laboratories that would want it worldwide. And as you rightly mentioned, Innes Laboratories with Yannick Koch and Felix Kalkmann in Switzerland is now doing the imaging flow cytometry method. It is it's totally easy, it's fast, it's reliable, and what makes it so nice, you put in the sample, you don't search under a microscope. It can't be said that you are cherry picking certain areas. Whatever you put in, you get out, and that is the result. And it's visual because it's the imaging flow cytometer. And so it's an easy modality. The PET scan on the other side, as you mentioned, I work with Jeroen Den Dullen. It's a very expensive endeavor. So this is a first attempt to search in the body from the head right through all the vasculature into the organ system on where these microclots are situated and the amount of damage they cause and how, if we can detect them. So we haven't done it yet. However, in his lab, in Jeroen's lab, we have set up the imaging flow cytometry method, the exact same method that Innes is using, so that they can screen patients before they take them to the PET scan to determine how the microclots look in the blood. And then hopefully we have an idea, the severity of the microclots in the blood, how it looks with the PET scan in the body.
It's also a very beautiful bridge, the analysis and the apheresis, because Innes has both of those. And it's just amazing how progressive you and the boys in Switzerland are. Thank you again.
Thank you.
Remember in the days when I got the first Zeiss microscope, the fluorescent one in Germany, we had numbers of 100% of the samples you tested were in long COVID cases were showing microclots, and about 80% in ME/CFS. Obviously, now you have been going through a much more broad patient population. Do you have any new numbers to share? Long COVID, ME/CFS, and other illnesses?
Yeah, so if we test long COVID patients with a diagnosis, so someone must have been new onset symptoms after acute infection, the typical long COVID profile. We have seen microclots in every single patient that we have looked at. It might not be as high as with alpha and beta and delta variants, but we do see microclots if they've got complaints of long COVID symptoms. If they do have, if the clinician feels that they've got vascular complications or endotheliitis, we do see microclots in those patients. Perhaps not as high as previously seen with the earlier variants. If we look at ME/CFS, they slap bang in the middle. So if you've got your control here, your ME/CFS is raised microclots, and long COVID is still more. So it is, it's halfway between the two. But on that note also, we also see significant platelet hyperactivation in ME/CFS and long COVID, to such extent that if you look at the platelets of these two populations, you cannot distinguish between the two. Definitely, they've got less microclotting. It might be due to the fact that ME/CFS, specifically before acute COVID, have got other viruses, so also post-virus syndrome, other viruses that cause or played a part in the disease that perhaps is not so virulent as the spike. But the dilemma is, what we also notice, many long COVID patients left untreated goes into typical ME/CFS symptoms, which is significantly cumbersome and worrisome for us, because how do you how can we treat, and we need trials, and that is the most important thing that we need to do.
Thank you very, very much, Sabine. Great. Let's bring in our panelists who will be happy, I'm sure, to quiz you on these questions. Yes. So go ahead.
Sabine, I'm still dizzy. If you ever need a job in Cyprus, we could treat 3 times more patients with you as an MD here.
I would love to set up a clinic. I'll be there. I'll teach the doctors, and we'll go on there.
Amazing.
Yeah, they call me Hurricane Hazen for a reason.
All right. Yeah. So first question: fecal transplant versus cotton ball transplant. Advantages, disadvantages.
What do you mean by cotton ball transplant?
In Europe, we have a technology where they don't use the feces, where they transplant, where they kind of clean the feces and they extract and put them on cotton balls.
Yeah, and they basically are giving the metabolites, right? I don't know. We don't have that much data, and here's the problem: why we don't have that much data is because, listen, in America, we're limited. You cannot do fecal transplant unless you have C. diff. And unfortunately, you cannot understand these microbes unless you have a validated assay to look at these microbes, and you have a proper protocol that you can kind of is similar, that you could say, okay, well, this is why I'm accomplishing something, right? My goal and my hope is to work with all these companies that are doing fecal transplant and to say, let's look at the microbiome. Dr. Barodi and he was doing fecal transplant in Australia, and I was looking with him. And so we started understanding what is a normal microbiome. But here's the thing: you can't really understand what is a normal microbiome until you've analyzed a lot of stools with a validated assay, and you kind of say, why is Alzheimer's looking like this? There's a signal in Alzheimer's. There's a signal in Parkinson's. There's a signal in a carnivore. There's a signal in a vegan, right? Because like I showed at the beginning, you cannot compare 2 samples. So if I'm seeing a signal when I'm improving, what I'm showing you, which is really, beautiful and all that in my own way of presentation, is also very skewed because I'm seeing what I want to believe of bifidobacteria, right? But there's so much more to the story than bifidobacteria.
Wonderful. So great. Thank you, everyone, for being here. You may notice on the screen, I will sometimes bring up the comments so you can see them real time. Many people are asking questions. Yes. And I was thinking, Marcus, is that when you guys are working with apheresis and you are removing, in a sense, microclots, you're removing autoantibodies. What seems to be interesting is when you remove it, the patient may improve, but they reform these patterns over time. So they have to continue having it done. What is it that you guys see in real time when you have taken out a lot of these inflammatory patterns?
Excellent question. So yes, as a mono treatment, as a standalone treatment, apheresis alone, no matter if that is help apheresis, inospheresis, or immune absorption, or a combination of the three, can only go so far. If you don't combine it with either hypothermia, nutraceuticals, especially molecuzan, or other therapies, where you also go after the root cause, apheresis would probably work for 40, 50, 60% of the patients, and they wouldn't relapse because they're going back to homeostasis and the body then can take over and recover. But for the other patient group that you just mentioned, multimodal combination therapy has been the only way that we saw working to achieve permanent results. And then there's a third group of patients. So let's say you come in a wheelchair, we bring you up to 65%. That is a great result, right? But then they come back and go like, what about the other 35%? And they come a second time, and we then add monoclonals in, monoclonal antibodies, IVIG, subcutaneous IgG, peptides. So literally, if you don't tackle this problem from all sides, your cards are quite bad. So again, there's a lot of monotherapies, but in our experience, if you don't combine therapy, sometimes even at the same time, you just don't have the results that you get by combination therapy.
Well, this leads right into Shankara from a clinical point of view. How do you know who needs what?
Well, let me come back to you, Philip, on our point of view. So what we've done, I currently have the feeling I see Brigitte more often than my wife. So we're working very closely on so many things. There's so much data interchange. Everything from science goes directly, at least here in our clinic, into treatment. So science-backed only, believe in the science. Second of all, don't hesitate, invest, create treatments. So literally, we started with a portfolio of one treatment. That was help apheresis 4 years ago. By now, we have a portfolio of over 30 treatments, right? And for me, it was very important to collect all of these treatments that are out there and test them. And the ones that didn't work for our patient population, we no longer offer them. Of course, everything has to be done under the do no harm principle. But there is a lot of treatments. Now, is that enough? Well, how do we know which treatment we use for which person? Now, in my opinion, we're already at 40 to 60% precision medicine. The tests of Brigitte, the tests of Risha, other tests, GPCR autoantibodies, the whole panels, the new labs that are now coming up. So there is a big part of the treatments that we can already target very well. But let's be very honest. For now, we're also having a part of the treatments where the good old pattern recognition. This treatment worked for a person with this symptom set. Let's try the same for the next person with the same symptom set, where that also has to be applied. And we're not shy of saying exactly, this is what we know, this is what we don't know. So here we are in the section of precision medicine with the treatments. Here, it's good old-fashioned pattern recognition. We don't know what's going to work for you. Let's throw things up at the wall and see what sticks. That is still a part of our clinical reality.
Listen, folks, I think this is brilliant. And what I want us to do as we close out, we're coming to that 2.5 hour mark. So I want everybody to think about what would their takeaway point be? What is it that they would want to say to our listeners now? I mean, we have almost 570 people live with us now, and more people will watch it down the line. What point would you want to give them based on your research, based on your experience? Excellent. And Marcus?
Well, my takeaway is very simple. My message is don't lose hope. I had severe long COVID 5 years ago, so I know how it feels to be looking at the ceiling 23 hours a day, being completely in pain, not being able to talk or even hold a glass of water. And me, and a year later, my mom, we recovered, and so many of us recover. So there is tireless people who work behind the scenes. There's new laboratories built. There is AI integrated. There is new tests developed. There is constantly new treatments. So yes, it is difficult, but there is some things that you can do at home. Inform yourself, reach out to the people that know how to treat it, right? And most important, share your story, because if we don't speak out, nothing's going to change.
Excellent. And Bridget?
Yeah, I have 3 messages. One message for other scientists. We have to be open-minded, as it was mentioned before. We have to talk with each other, and we have to work together. My message to the therapist is, the therapeutics and medical doctors is, we have to go from two sides. I learned it. From one side is from the science, but we need also the experience from the medical doctors. We must know what is helping, and then we have to think what could be the mechanism or the pathophysiology. So we need this interaction. The third message for all others is, yes, there is hope. And I have to agree to Marcus, what he told, inform you and take care of your body.
Excellent. Thank you, Bridget. And Rezia?
Yeah, I think the important thing for me is that we can never lose hope that there will be some sort of respite soon. I think there are researchers and clinicians working around the clock that don't sleep, that are constantly thinking of how to plan new research, how to look into new frontiers. So for the patients, never to lose hope, because there are people looking out for you. And then I think the most important thing for a message to me is protect yourself. If you're on a flight, wear a mask. If you go out to a function where there are hundreds of people that you don't know what their infection status is, protect yourself. That's the only thing that you have in your hands to try not to get reinfected. We are only one infection away from long COVID, every single one of us. And we don't even know what the underlying effects is. Even if you've got an infection and you are asymptomatic, down the line you can develop long COVID. So I think the most important message for me is protect yourself and look after your family.
Wonderful. And Sabine?
Well, I'm gonna say, as the girl that basically went on airplanes without a mask and somehow managed to survive, I'm gonna say, calm down your body and stop panicking, and see the light and see the hope and see the lessons we've learned from COVID, the lessons on the microbiome. And I think the microbiome to me is about, calm, zen, decrease stress, decrease anger, decrease caffeine, decrease alcohol, decrease the stress, decrease the fear, decrease the anger, and stop the division. Unite to talk together, bring on discussions as scientists to see both sides of the narrative, not just one, always trying to remain as unbiased as possible, so that we can advance science, because it doesn't help us if we fake the science or change this or trick the science. It doesn't help us. We're eventually all going to become patients. To me, it's about a mission to preserve the microbiome of humanity. We've killed too much. We need to build now. We need to bring back microbes. And I think with that is really peace, calm, unity, stop the fear, and go play in the garden. It's summer. All right, that's my message. Thank you.
Wonderful. Thank you all very much. And I know we've got a lot of people with us today live, and I know a lot more people will watch it afterwards. Remember the science. Let's all work together to make a better future. I'll ask my guests to stay with me until after the outro, but for everyone else, have a great evening.